Hello, my name is Kazi Iqbal. I'm a graduate from Centennial College with an advanced diploma in biotechnology and industrial microbiology. During my 3 years of study, I took part in a biotechnology co-op program where I worked at the Centennial College laboratory, where I worked as a lab technician. Part of my job was to create subculture microorganisms from cryogenic vials, such as Streptococcus, Lactobacillus, Shigella, Salmonella, and much more. My work term at Centennial College as a laboratory technician has made me realize and convinced me that I would like to pursue this as a career and, most importantly, advance in this field of study. So one of my favorite courses in Centennial College was Microbiology Project, where students were given 4 months to conduct an experiment of their choice under minimal supervision. The students then presented their data in an oral and written manner. So in my last semester of study, I conducted an experiment in determining E. coli using conventional microbial methods compared to PCR methods using various water samples. So how I conducted my experiment was I filtered water through a membrane filtration unit in a 0.45 microunit filter paper. Then I took the filter paper and I let it grow on FC agar. I chose FC agar because it is selective for fecal coliforms and it will give me green sheens. After that, I let it grow for 24 hours at 37 degrees Celsius to get growth. So after incubation of 24 hours at 37 degrees Celsius, I selected the green metallic sheens and further subcultured it into a Fisher tube containing media. I split the Fisher tube into two, and one sample I used for biochemical testing and the other sample I used for PCR. For the biochemical testing, I used TSI sim citrate to identify it. So after running the gel for 45 minutes at 150 volts, I received these bands. As we can see, the creek water and the toilet water displayed bands, whereas the tap water had no bands. So in conclusion, in comparing the conventional microbial method to the PCR method, showed the sensitivity in detecting E. coli of the 3 samples were the same. But in terms of time, the PCR was much quicker, taking only 24 hours, whereas the conventional microbial method took 4 days. I want to thank you for your time. I hope I have demonstrated the necessary laboratory skills that can be incorporated into your organization.